Hello everyone. Today we will discuss about lipid metabolism. Lipid metabolism is a series of complex biochemical reactions involving the digestion, absorption, synthesis, decomposition and transport of lipids. Acetyl-CoA is the central molecule of lipid metabolism. Let's see the sources and fates of acetyl-CoA. Acetyl-CoA is derived from pyruvate by the action of enzyme pyruvate dehydrogenase. This pyruvate is the end product of glycolysis. Acetyl-CoA is also derived from carbon skeleton of some amino acids. Triacylglycerol, which are stored in the adipose tissue, are broken down to fatty acids and glycerol in fasting and starvation. These fatty acids are oxidized to form acetyl-CoA. So these are the sources of acetyl-CoA. Let's see the fates of acetyl-CoA. In well-fed state, acetyl-CoA undergoes TCA cycle. Reducing equivalents are generated, which enters electron transport chain and generate ATP. Next fate is fatty acid synthesis. Fatty acids that are synthesized are stored in the form of triacylglycerol in adipose tissue. Acetyl-CoA is also the substrate for cholesterol synthesis. This is in well-fed state. In fasting and starvation, this acetyl-CoA formed from lipolysis and beta oxidation from triacylglycerol, there will be formation of ketone bodies. So these are the sources and fates of acetyl-CoA, which is the central molecule of lipid metabolism. Let's see the major metabolic pathways in lipid metabolism. In well-fed state, first one is digestion and absorption of lipids. Second one, de novo synthesis of fatty acids which occurs in cytosol. Then synthesis and storage of triacylglycerol. Synthesis occurs in smooth endoplasmic reticulum. Lipoprotein metabolism. Cholesterol synthesis, that is in cytosol and in endoplasmic reticulum. Then compound lipid synthesis, for example glycolipids and phospholipid synthesis, which occurs in smooth endoplasmic reticulum. So these are the major metabolic pathways in well-fed state. And this is regulated by the hormone insulin. In fasting and starvation, first one is lipolysis, that is triacylglycerol breakdown, fatty acid oxidation, which is in mitochondria, ketone bodies synthesis and utilization, that is also in mitochondria. These are the major pathways in fasting and starvation. This is regulated by hormones glucagon and epinephrine. Coming to the metabolic pathways, it starts with acetyl-CoA, which is derived from pyruvate by the enzyme pyruvate dehydrogenase. This acetyl-CoA enters TCA cycle, but this acetyl-CoA cannot cross the mitochondrial membrane. So it combines with oxaloacetate to form citrate. Citrate can now cross the mitochondrial membrane, so it enters cytosol. In the cytosol, citrate is broken down into acetyl-CoA and oxaloacetate. This acetyl-CoA is involved in fatty acid synthesis, and these fatty acids are stored in the form of triacylglycerol in adipose tissue. Acetyl-CoA is also a substrate for cholesterol synthesis. This is in well-fed state. In fasting and starvation, triacylglycerol is broken down by lipolysis to form fatty acids and glycerol. These fatty acids are activated to form fatty acyl-CoA, which undergoes beta oxidation to form acetyl-CoA. And there will be synthesis of ketone bodies from this acetyl-CoA. Coming to the transport of lipids, because these lipids are insoluble in water, they cannot travel through the blood alone. So the body uses specialized transport vehicles and pathways to move them between intestine, liver and peripheral tissues. The primary method for transporting lipids is through lipoproteins. These lipoproteins are spherical particles with a hydrophobic core containing triglycerides and cholesterol esters and a hydrophilic outer shell made up of phospholipids, free cholesterol and proteins called apolipoproteins. These lipoproteins include chylomicrons, VLDL, LDL, HDL, etc. So this is about lipid metabolism. This is today's topic. Thank you for watching.